Good morning, everyone, and very welcome to all the people that's here in person in the room and to everybody joining us on the virtual platform. It's really an exciting day to see so many people present in person again, representing a lot of different countries all over Africa and also internationally. Welcome to our US delegates that had to really travel far to get here. And then welcome to all the South Africans that's also here today. So I'm going to start the workshop just to give you a brief overview of where you are at University of Pretoria and where everything fits in. And then I want to speak a little bit about SABRENet and the DITRA project, and also present to you what this workshop is about. It's a workshop, so the word work is quite important. It's not just sitting around listening to presentations. We really want to have some outputs at the end of this workshop. So firstly, welcome, and we're going to get to know everybody and through the teas and the lunches and all the social events as we go forward. Louis, you can load my presentation. Oh, that's Kendra. I'm the other bat woman, but that's not me. Sorry, I think I just called it Markotter or something. No, it's a PowerPoint. Okay, just give us a minute. With some of the initial slides that you don't have to really see for the sake of time. So you're at University of Pretoria. We in Pretoria, most of you that's here know where we are. It's in the middle of South Africa. It's our administrative capital. And it's a university that's ranked among the 100 best universities, specifically in some of our science disciplines. We've got over 55,000 students. So we're quite large, over several campuses. This is one of the campuses. We spread over the whole of Pretoria, starting from here up to the north of Pretoria, where we have our veterinary campuses and our other campuses. What's quite unique about the University of Pretoria in the South African environment is we're the only university with a veterinary faculty. So we're the only university training vets. Now, if we talk about biosurveillance, coronaviruses, One Health, that fits very well into the picture. We've also got a School of Health Sciences, Faculty of Health Sciences, where we train our medical doctors, and then all 9 faculties that's from social sciences to economics. We've got a very good business school that also do a lot of training throughout Africa. So we really function in this One Health environment. Now, in this environment, we've got certain centers, and the Center for Viral Zoonosis is one of our flagship centers that's really, okay, there's my slides. Now I can go to, it's not moving. Okay, there we go. Just a few pictures. So it's unfortunately winter now, but if you're here in the spring, summer, you would see these purple trees all over Pretoria, and that's why it's also called the Jacaranda City. And apparently, if one of those flowers falls on your head, you're going to pass your exams. So, but it's not very difficult because there's flowers falling the whole time. So, but you feel lucky after that. So we produce research. We're a research-intensive university. And we also train students up to PhD level. So that's quite important, and that fits also very well into this workshop. As I said, we train the countries, we're the only institution training veterinarians. So our Faculty of Veterinary Science is on the northern side of Pretoria. If you've never been there, it's very close to the Onderstepoort Veterinary Institute. That's our animal health institute. So it's just opposite the road of them. And then getting to the Center for Viral Zoonosis, where I'm based. It's based in health sciences, but it's really an initiative in UP across all the faculties. So although I'm the leader of that center, and my work is very much focused on bat pathogens and a bit of rabies and a few other things, there's lots of other people also associated with this center. That's not just bat pathogens. So we do a lot of arbovirus work, we do some wildlife work, we do rabies. There's a lot of other programs that's not so relevant for this, but maybe relevant in the bigger picture. In that center, I'm also in a South African research chair. And what this means is I have a funded project actually by our government, our Department of Science and Innovation, who contribute towards my salary, but also towards research costs. And these run in 5-year cycles. And it's specifically to look at small mammal zoonotic pathogens. And then my group has got a specific name, Biosurveillance and Ecology of Emerging Zoonosis, that functions in the center. So we do a lot of different things. I'm not going to go into this in detail, but we have surveillance and diagnostic capacity building. So as a lot of you that's in the room will appreciate that we really need to look at diagnostic methods or surveillance methods that works in our countries. We can't just always implement very fancy things. We really need to look at low resource things that works for us. It must be affordable, it must be available. So we do a lot of that work. We also do some more fancy things that's not just surveillance, where we really start looking at host pathogen interactions. We follow the One Health approach, and we're going to talk about that quite a lot in the next few days. So we also need to look at interactions that's causing risk. And Ava from the US is going to tell us a lot more about that, where we look at human-bat interactions on several interfaces. And then the ecology. There's a lot of ecologists and wildlife people in this room, which is actually great, because that is extremely important if we start looking at biosurveillance of pathogens. It's not just about the pathogens anymore. It's about a lot of other factors. Then you're on Future Africa Institute campus at the moment, and I'm also in a chair on this campus, really specifically focusing on One Health. So we're trying to facilitate all the One Health initiatives in the university, but also broader in South Africa. And the aim of this institute, you will see there's no labs here. It's really a collaborative space where we bring people together with the focus on Africa. We're very pan-African, but also within the university across faculties to facilitate transdisciplinary research. So it's really going that next step, not just interdisciplinary, transdisciplinary, getting government involved, communities involved, everybody around the table when we talk about research. And that's where you are now. So just a bit of background. I think it's always just good to see where we are, and for the people who's joining us virtually also, you know, where all of this fits in. So coming to this workshop, this was initiated originally by the International Corona Network from the UK, who is funded by Biotechnology and Biological Science Research Council, as well as DEFRA. And I'll say a little bit more about what this is about, but then this became bigger because of all our other collaborations. So we included the South African Science and Innovation, which is my research chair, the NRF, and then also the Defense Threat Reduction Agency of the United States of America. And the link here is because our project in South Africa is very well funded by them. So they play an important role in that. Oh, now I'm getting out of breath. And then EcoHealth Alliance, there's quite a few, 2 of their people is actually is one of our major partners. So they were also part of arranging this workshop. So going to the UK ICN network, I think it's important that we just have a look at where this fits in. So there's a website, and you can go to that, and there's actually a bottom here, there's a link that you can click to join. It's open for anybody to join, so you're welcome to go and look at it. And there's several international, there's an international management committee, but it also includes people from all over the world, including Africa, that's linked to this project. And what is the aim? It's a network. So it's really to provide a network of networks with including UK partners, but also other international bodies and other international partners. And it's really to facilitate and coordinate interaction, like we're doing with this workshop, in the animal-human environment interface. We can again come back to One Health. It's not just looking at viruses, it's not just looking at ecology, it's really this whole interface. Looking at ideas, discussing it around tables, create research opportunities and build evidence-based roadmaps for One Health research. And that's what we're going to do in the discussion groups. And really looking past SARS-CoV-2. You know, that we need to start looking at what do we need to do in terms of prevention also going forward. And then disseminating knowledge and how to use that knowledge. So that you can go and look at their website, but Professor James Wood will also now give a presentation, who is one of the leaders in this network initiative. So coming to Africa, because that's what our main aim with this is, and this is a map that Dr. Geldenhuys has provided me with. Why are we here? So if you look at this map, this summarizes in terms of coronavirus data that's out there, what we know. And you can see we've done quite a bit in South Africa, but in the rest of Southern Africa, and that is what we focus on here, is the SADC region, the Southern Africa Development Community. There's a lot of countries where we know very little. So we really want to strengthen biosurveillance, firstly on coronaviruses, but obviously the principles can be applied much broader. It's not just about coronaviruses. And really color in this map a bit with information on countries where we have absolutely no information, and use the strength that we've built up in South Africa to really work in the region, build capacity, and get sustainable programs off the ground going forward. So Marika is going to talk much more on coronaviruses and the detail around this, so I'm not going to go into that. So why are we here, and what is the aim of this specific workshop? It's firstly to bring people around the table and build collaborations and networks. It's definitely not everybody that's here. We know that. We try to put together a group that we can manage in a very short time. We need to bring all the other people also to the table and then do more workshops in the future in countries or in different regions to get more information and more people involved. We really want to look at assessing the gaps in research of zoonotic bat coronaviruses, so we're going to focus a lot on coronaviruses. But like I said, it is a bit broader. And then assist country representatives with a strategic plan to look at implementing in-country surveillance in bat populations. Not just bats, we also need to look at all these other interfaces, although that is the focus. And we really need to look at what's the gaps. We can't just go in guns blazing and do things if we don't know what our gaps and our limitations are in some of these countries, and the needs and the requirements. And very important, transdisciplinary One Health approach. So, as some of you are aware, I'm the co-chair of the One Health high-level expert panel, so I always have to show this slide. If you haven't seen it yet, this is the new One Health definition that's been adopted by the 4 UN agencies that was developed by OLAP. And it says basically the same sort of thing that we previously said in terms of One Health, where it's really about healthy ecosystems, healthy animals, healthy humans. But this circle is also really important. Who should be involved? Society needs to be central in things that we do going forward. We should move the focus to really understand what we need to do to change things. And that involves quite a lot of communities, local, regional, global, and a lot of sectors and disciplines. There's a lot of underlying principles associated with this definition. Equity, sustainability, those words, extremely important. We hear it a lot, but I don't think we always think about what it really means and how we're going to achieve it. So this is something that we can build on during this few days also, thinking about this picture with everything we do. Who needs to be around the table? So how are we going to do it? So we did a pre-workshop basic information gathering plan where we ask you to fill in some details specifically for the SADC countries, and we're going to build on that. That helps us to identify needs, gaps, and it's already quite interesting the type of info we're getting from that, where we can see there's a lot of basic capacity building that we need to still do before we just go in and start with large biosurveillance programs. We're going to do that first starting with specialist lectures, and that is what's going to happen in the mornings, and our virtual audience is also joining us for those lectures. And there's some of these lectures that's going to be presented by the older research generation that's experts in the field, but we really wanted to bring in the emerging scientists. So a lot of these lectures are going to be from people who's actually working on the ground on some of these aspects. So they can really speak practically about their experiences and their knowledge. Then in the afternoons, and unfortunately our virtual audience is not going to join us for that, but we will make the results available, we're going to have working group sessions where we're going to work in country groups really to look at developing surveillance plans. And we hope to have some drafts ready at the end of the workshop. We need to look at gaps, so we will do some gap analysis. And then the outcome of the workshop will be a white paper format that we're all going to contribute to in terms of what's going on in the SADC region and what's the things that we want to highlight in terms of coronavirus biosurveillance going forward. Obviously, if you think about this, what can we do with this? We can start looking at how we're going to fund things. So a draft plan on the table can be developed into funding opportunities and being submitted to that. So that will also be the aim. I don't have a watch on. What's the time now? Okay, so I still have some time. Maybe just raise your hand, Marika, when I'm out of time. So I'm going to just talk a little bit about SABRENet. So SABRENet is this little logo, and you will see it's on your T-shirts and everywhere. And it stands for the Southern Africa Bat Research Network. So originally when we started with a funded project by DITRA, we started doing it under this network. So it's really a small group that originally started, and you can see the partners on this slide due to the funding opportunity that we had. But the idea is really that SABRENet is the whole SADC region. And that today is the start of really bringing people together in this region and from the different countries. So we have our international partners, which is Uniformed Services University, that assists us a lot with serology assay development. We have the National Institute for Communicable Diseases. That's also our only BSL-4 lab is in their facility, and Jacqueline Weyer is here that's representing them. We've got Eco Health Alliance that you will hear a lot about during the morning talks. University of Eswatini, who's also going to talk. Ara there in the back. Titsong National Museum of Natural History, where we do all our bat taxonomy work in collaboration with them. So they also represented. And then all our government departments that's relevant to this project, which include environment, agriculture, as well as health as part of the project. And then you will see two flags there. I don't know, the people whose flags it is will recognize it, but a small part of the project we were able to bring in Mozambique and Zimbabwe already. So there's a lot of representatives from those countries here today. And although we couldn't do as much in those countries, they're at least part of the project with the idea that we can expand it going forward. So what is the overall objective? It's not just about coronaviruses. We're looking at the high consequence pathogens, which is the phylos, the Nipah, and then the coronaviruses, and evidence of spillover in humans and livestock. So this is what this project is about, but we're also doing some other work with other funding opportunities where we look a bit more broader at wildlife and wildlife risk and a few other things that's not specifically linked to this. First, it's about capacity building, workshops like this, where we really want to start with talking, but then we need to do hands-on training on certain aspects also. And we've done quite a bit with Botswana, with Eswatini, where people has been spending time in our labs, really learning techniques. Biosafety is always very important. Then we really want to establish, and we've done that, and you will hear a little bit about that, on serological and nucleic acid detection platforms in South Africa firstly, where we look at human and animal health labs together with the university. So we're not just building capacity in University of Pretoria. it's really important that this translates also into some of our public health institutes and animal health institutes. And then we go out and we sample and we test and we test and we test. So we do that and we try and see what we find. And we use all kinds of fancy methods to try and look at risk. We're also doing human behavioral risk studies that we're going to start with soon. It was only in year 3 of our project. And then we do a lot of multi-stakeholder workshops. So this is really what this project is about. It started 2 years ago. It's a 5-year project. So it's still running till 2025. But the idea is really to use this as a base to do more countries in SADC and get more people involved. So I'm not going to go into detail. We're going to discuss all of this later today, but there's a lot of things that you need to consider with biosurveillance, and that's what we're going to discuss. It's how are you going to get the samples? How are you going to identify the bats? What are you going to do with the data? How are you going to test for the pathogens that you want to test for in terms of serology as well as nucleic acid assays, all your environmental and ecology data? Is it available, or do you need to go and try and get it for your region? You know, those things are also important. The animals are important, the biology of the bats, livestock, other wildlife, all those things are important. Movement, contact with people. Is there a risk? In a lot of the studies that we've done all over Africa, we would go and test bats in the middle of nowhere where there's probably very little risk of them ever getting into contact with people or other livestock. We really need to shift to target our surveillance where there's risk so that we can really do something with the data. And then it's all human behavior studies, very important. And it's overall, there's capacity building. We need to look at how we report these things. That's quite a hot topic internationally also now. How much should we share? How should we share? Should everything be always available in real time? We can also have those discussions. And looking really at how we can reduce risk before we have another COVID pandemic. So it's really about trying to mitigate risk. And communication and outreach, Tetson will also talk about that later, extremely important. And the way that we target that communication and outreach, I think it's so important that it's targeted right. COVID showed us that. As scientists, we communicated very badly during the pandemic and too late. So it's really having that that base that's already there with communities to communicate in the right way. So we sample all over South Africa, and then the plan is later in year 4, we will move a little bit into Mozambique and Zim to do actual sampling, although those countries have been part of the capacity building up to now. Just a few pictures. So you're going to see some of these pictures a lot. You know, it's important that you've got biosafety and biosecurity expertise. It's important that you've got certain infrastructure. Everything needs to be done safely, and we'll talk a little bit about that. It's also important that you know how to sample the animals without causing harm or harm that's really going to be detrimental to them, and doing it according to ethical and regulatory standards. So we can also talk about that. Museums, very important. Taxonomists, very important. Bat biologists, extremely important. I think Ara will highlight that also in some of his talks. So in terms of coronavirus biosurveillance, I'm not going to talk about all our other pathogen data, and Marika will talk about this a bit more. We've done a lot of sample testing, and we get what other people get. Crazy diversity in terms of coronaviruses. And she will highlight a bit more what is important in this diversity that you're getting in terms of nucleic acid sequences. But we do get some that's potential zoonotic. We do get SARS-related viruses in the bats if we test enough horseshoe bats in Southern Africa. And there's also a few others that she will talk about. So there's relevance in getting the nucleic acid sequences, but this is definitely not the only thing that you can do. This is just the first step. And we do longitudinal surveillance, so we do it over time, and this is some of the work that she will also highlight that's quite important. You need to sample regularly to look at patterns of excretion, try and link that to environmental or other risk factors, try and link it to the host biology. If you just go in and out, then you're only going to have that diversity tree, and that's all it's telling you. There's lots of coronaviruses, and you're not going to have any other data. So this is some of the work that she will highlight, where we've looked at high-risk periods in South Africa because we were doing monthly sampling over years, and that's hard work. It's not, you can ask my people, they're more in the field than in the lab, and then they're also the lab people. So it's not like we've got separate teams in terms of field and lab. So it's really big teams and lots of capacity that you need to do this. Just something I'm going to mention, because this is quite important if we talk about biosurveillance. Yes, you can do nucleic acid detection, which is your molecular methods, and you can target specific sequences with that. But for certain studies, you need to look at serology, especially if you start looking at spillover hosts. There's not going to be an infection at that point. You want to see if there's exposure, so you want to look at antibodies. And our wildlife serology tools is actually not very well developed. We don't know cutoff values and all of that. So we're really working on this with this project to see how we can improve it by using a multiplex Luminex system, which is bead-based. So you can throw 2 microliters of serum in there and test theoretically 400 pathogens. And that is what we need to start moving towards, but we need to understand the limitations of those assays and how we can interpret the data. So we'll talk a lot about that also. Just a few slides. I'm not going to go into detail, but we do look at movement patterns. Lau de Vries will talk about that more and the tools that you can look at. Because how are you going to determine the risk if you don't know where the bats are going? You know, are they feeding close to people? Are they moving large distances? What's their pattern? So this is actually showing us a lot of nice data in terms of risk, combined with camera trapping and all kinds of other things to see what other wildlife is in the area. Hotspots, this is some of the work that Mariette will also talk about, you know, where we're really trying to see overlaying our pathogen data with human population data, land use changes, where our caves are, all that data, environmental data, and see if we can determine risk. This goes back to hotspot mapping, but really using local data, not extrapolating from global data or modeling risk. So we're really trying to move towards that, and you're going to hear quite a bit about that. And then the natural history collections, where we have a very, very close relationship with the museums and with the bat taxonomists, where we really add through our project data that's important for conservation, species distribution, and it's somewhere where it's going to be sustainable. It's not in a university research project, and then when the project ends, it's not there anymore. So we really try to contribute to the museum collections and to the data so that it's there for the long term. Education and communities, you will hear a lot about this, but extremely important to do this. And one thing I'm going to mention is 2 minutes. Thank you, Marika. So we developed an extensive standard operating procedure manual between all our partners. There's 2 volumes. It's a lot of pages. Everybody that's part of the project contributed to that, including our government. So we actually standardized our PPE requirements and things like that in agreement with what our regulatory requirements are. So we've done all those things, and these manuals is there to use in the SADC region or even broader. It's very specific for Africa. We really revised some of the previous predict things so it works for us. And we're not going to, in this workshop or in the presentations, talk about the SOPs in this. We're going to give you a broad overview, but this is going to be available if you need the SOPs. If you need to know how to do the test, if you need to know how to capture the bat, this is going to be available, and we'll make that available to everybody. We're revising it as we go on. We're adding to it, but I think it's a really nice document that we have to start from. I'm now looking at Kendra. You actually did quite a lot of work on this. And then Marika will talk about this. A while ago, we did a review on what we think should happen with coronavirus surveillance in Africa, summarizing what has been done, and she will, again, just highlight some of the points out of this paper. So coming back to what we're going to do. So we're going to listen to a lot of talks today. Some of them is going to be recordings from some of our international collaborators that couldn't be here in person. And then this afternoon, we're going to talk. And while you hear these discussions this morning, or the presentations, I want you to think about a few things for this afternoon. Firstly, why should we do this? Because if you're not convinced that this type of project is important, it's going to be very difficult to keep you motivated, because it's hard work. So firstly, think, is it necessary? Is this what we should do? You know, should we have these biosurveillance projects that's really focused on the prevention of future pandemics? Is it important? What should be in a project like that? So you can start making notes while people talk. What is the aspects of a project like that? And then who should be around the table? Where's your bad taxonomist? Do you have somebody like that in your country? Or don't you? You know, those things we need to start highlighting, and we're going to do that in the working groups today when we start discussing things in detail. But think about it when you see the presentations this morning and you listen to it. We'll share the presentations. It's also going to be available as recordings afterwards. So you will have it available to go back to it. But think about what you're going to do in the afternoon session with the information that you're getting, and thinking about your country specifically and your situation on the ground, because it's going to be different.